Okay, good afternoon, everyone. It is great honor to welcome you all to the 57th Korean Academy of Science and Technology International Symposium. I am Kyu Kim from Yonsei University. I'm going to chair this session. So let's get started. The symposium with opening remark from our president, Jin-Ho Chung. Jin-Ho Chung, President, please.
Good afternoon, distinguished colleagues and guests. On behalf of Korean Academy of Science and Technology, it is my great honor to welcome you to International Symposium and today's in-depth discussion on GLP-1 therapeutics. This is a top-notch science symposium. We have gathered pioneers who have advanced our understanding of incretin biology from receptor signaling and neural circuit of appetite to polyagonist and next-generation delivery system. Thank you for bringing your latest insight to Seoul and to the local and global community joining us here and online. After the talk, we'll have an in-depth discussion. We'll keep it clear and practical, focusing on three simple questions. First, where we stand in Korea. What capability do we already have in basic research, manufacturing, and clinical trial? What are the smart next steps to close the gap? Secondly, what do we see in the clinics? Real case and real outcome, who benefit, how much, and for how long? Thirdly, how basic science become a big impact medicine. Step by step, how does a lab discovery grow into a blockbuster drug and sometimes even a Nobel Prize level contribution? The GLP-1 story began with a simple question about physiology. Careful work over many years tuned those questions into treatment that help millions with obesity and cardiometabolic disease. As a national academy, we have one more goal: inspire the next generation. To the students and future scientists here and online, the breakthrough you hear about start with curiosity, good idea and data, and teamwork. You can do this too. Ask clear questions, share results, and work with others. That is how science changes lives. Finally, my thanks to our speaker, chair, panelists, and interpreters who make this program accessible to all. I wish you a lively and constructive discussion, clear and data-driven and people-centered. Welcome and enjoy the symposium and in-depth discussion. Thank you so much.
So now we are moving to the session one. The title is GLP-1 Research and Therapeutic Innovation in Metabolic Disease. So we will have 5 distinguished leaders in incretin biology in this session. So let me briefly explain this session because we are very tight time frame. So each speaker will have 20 to 30 minutes, and we will not have Q&A session because we will have discussion time in following session. So each speaker can use all the time for their presentation. Okay, so let's move on to first speaker. So first.
Hi there, my name is Daniel Drucker, and I'm a senior scientist at the Lindenfeld-Tannenbaum Research Institute in Toronto and a professor of medicine at the University of Toronto. And I'm delighted to join the CAST meeting and give you a sense of how I view the mechanisms of GLP-1 action, which has fascinated me for 40 years. So these are my disclosures. I work with a number of companies in the field, and some of them support the basic science work in my laboratory. So these are the medicines that we have today in many countries around the world, including Korea. They generally act through the GLP-1 receptor, but as we will discuss, the newer medicines such as tirzepatide also engage other receptors. And the two most commonly used medicines today are once weekly semaglutide and once weekly tirzepatide, approved for both type 2 diabetes and for obesity. Now, there are lots of medicines, including some from Korea, that are being developed that are to be taken as tablets. We already have oral semaglutide available, and there are multiple small molecules. Some have preferential residence time in the gut. They have different pharmacokinetic properties. And there's some question about whether there's any advantage of delivering GLP-1 medicines through the gut, because as we know, GLP-1 is made in the gut and does go first to the liver, where there may be some GLP-1 receptors in some neural structures in the portal vein. But here I've plotted for you, and this is actually data from thousands of people who either got oral semaglutide in the Pioneer program or injectable semaglutide in the Sustain program. And every single thing we measure, here I'm showing you hemoglobin A1C and body weight on the right, but everything that we see that we can measure is the same with oral versus injectable semaglutide as long as we correct for the circulating blood levels. So I think from the available data, it's really the blood levels that determine the pharmacodynamic outcomes. So why are we still talking about GLP-1? It's more than 40 years since we and others did the original discovery work. And part of the reason there's such excitement is shown here. This peptide has actions on many different organs. And very importantly, these actions are generally beneficial. The direction of the actions that you see would help people living with end organ complication associated with type 2 diabetes or with obesity. So we had approvals for type 2 diabetes in 2005. We had approvals first for obesity with liraglutide in 2014, and about 5 years ago for semaglutide, about 2 years ago for tirzepatide. But now we've had all of these phase 3 trials, including cardiovascular outcome trials, peripheral artery disease, kidney disease, arthritis, heart failure, liver disease, sleep apnea, and GLP-1 medicines have shown benefits in all of these indications. And we're starting to see expansion of the use of these medicines to help people living with these different health challenges. So this was the first set of trials with once weekly semaglutide, the STEP trials, that really showed the potential of GLP-1 medicines to produce weight loss that we had not seen previously with any other medicine. Of course, bariatric surgery is very effective at producing substantial weight loss. But prior to these STEP trials, we hadn't seen this type of weight loss with any other medicine. In some trials, 40% of people are getting 20% or more weight loss. And this started to happen really around the time we got COVID-19, and the news really did travel far and wide. So how does this happen? Well, GLP-1 produces satiety and people eat less. But there's tremendous inter-individual variation in the results we see. In some trials, people will lose 30% of their body weight with semaglutide or tirzepatide. And in the same trial, some people won't lose any weight at all. And when we stop the medicine, some people gain back all of the weight very quickly over months, and other people don't. And so how do we explain this? And so far, it's been difficult. There have been metabolomics, proteomics, microbiome studies, and here I'm showing you an illustration from genetic analysis. And we really have been unable to use genetics or biomarkers or omics to identify what underlies this variability in response. Now, some of the actions we understand very well. And for example, the first actions of GLP-1 were really to demonstrate that GLP-1 stimulates insulin secretion. Those are experiments that I did over 40 years ago in Boston. And we know there are GLP-1 receptors on beta cells and some on delta cells that make somatostatin and some on alpha cells that make glucagon. So the intra-islet biology regulating islet hormone secretion, it is quite well worked out. We never have a perfect response. There are still elegant studies being done to tease this out in more detail, but we know that this is where the action occurs for the glucose regulatory aspects of GLP-1 action. Now, some people have invoked a role for brain GLP-1 receptor signaling, including some scientists in Korea. And it is true that if you block GLP-1 receptor signaling in the brain with an antagonist, you will perturb glucose homeostasis. However, I view this, as somewhat of a modest effect overall. We restored the pancreatic GLP-1 receptor in GLP-1 receptor knockout mice. There was no receptors in the brain, but glucose homeostasis completely returned to normal, both the physiological and pharmacological response to GLP-1. So it may well be that brain GLP-1 receptors fine-tune hepatic glucose production and some islet hormone responses, but you don't need those receptors to get the pharmacological response to GLP-1 and lower blood sugar. Now, I'm showing you this study, which was published in Cell Metabolism in September. This was a collaboration with my lab and Dave Hodson's lab. And from time to time, we take a look at the available antisera that are used to detect the GLP-1 receptor. And in this paper, we studied 3 antisera with a particular focus on this one, AGR-021, which has been used in papers published in Nature, in Cell Metabolism, in more than a dozen, very interesting papers. And unfortunately, this antibody does not recognize the GLP-1 receptor. And some of you have heard me talk about this before. We've been, verifying the sensitivity and specificity of these antibodies for almost 15 years, and there's still a problem in the field. Why am I mentioning this? It's very difficult to read the literature and truly understand what's correct and what's not correct unless you yourself have used these antibodies and validated the antibodies. So you have to be a little bit cautious in taking at face value where the receptor might be based on the use of reagents that are not validated. And we did experiments with this antibody to use it for Western blotting, immunocytochemistry, fluorescent activated cell sorting. The bottom line, unfortunately, is that it's useless. Actually, it has no sensitivity or no specificity for the GLP-1 receptor. So among the most important and most exciting actions of GLP-1 beyond glucose control and weight loss are the actions in the cardiovascular system. And this is a meta-analysis published in June in Diabetes Care, where we now have about 11 trials in people with type 2 diabetes and 1 large trial in people with obesity. And it's very clear now, if you have a long-acting GLP-1 medicine, you're going to see a reduction in heart attacks, strokes, and cardiovascular death, and probably even all-cause mortality. But how does this happen? We still fully don't understand all the details of the mechanisms. So it's logical to say, well, it must be secondary to control of blood sugar and body weight. And maybe they help a little bit, But here's a really instructive cardiovascular outcome trial with a long-acting GLP-1 medicine, albiglutide. This medicine was pulled from the market by GSK because it wasn't commercially successful. The hemoglobin A1C reduction was maybe 0.6%, 0.8%, that there was almost no body weight loss, and it could not compete in the marketplace with the more effective medicines at the time that included liraglutide and dulaglutide. But ethically, the company continued the cardiovascular outcome trial. And this was the Harmony outcomes trial. And the results are shown here. There was a 22% reduction in MACE, heart attack, strokes, cardiovascular death, with no weight loss and minimal hemoglobin A1C reduction. So this really reminds us that it's unlikely that hemoglobin A1C reduction and body weight reduction are major contributors to the benefits we see, certainly for atherosclerotic cardiovascular disease. So could it be the receptors in the heart? Well, let's take a look. We identified that there are some atrial receptors, GLP-1 receptors, and some endocardial, endothelial GLP-1 receptors. And using mouse genetics, we can knock them out. And then we can carry out ischemic cardiac injury in mice with and without administration of a GLP-1 medicine. And when we do that, we no longer see cardioprotection with liraglutide. So we can get pretty excited and say, hey, maybe those receptors in the heart in those locations are contributing to the cardioprotection in the heart. But if we then look at human hearts, we see that the receptor localization is different. It's not really in the same endocardial endothelial cells, but some cardiomyocytes in the normal or ischemic heart in humans express the GLP-1 receptor. So are those receptors on some of those heart cells responsible for some of the heart protection? Maybe. It's just more difficult to do definitive experiments to demonstrate whether or not those receptors are key. One could do those in isolated working human hearts, and that would certainly be an exciting experiment to scrutinize. So what about the kidney? We have excellent data showing that in people with type 2 diabetes, if they're treated with a long-acting GLP-1 medicine, such as semaglutide, that's what was done in the FLOW trial, there was reduced progression to end-stage kidney disease and reduced cardiovascular death. So how does that work? Maybe it's a direct effect on the heart and the blood vessels in the kidney. Maybe it's through control of blood pressure. How does GLP-1 lower blood pressure? There are some studies that suggest it's through the GLP-1 receptor on endothelial cells. In these unpublished studies, we took a look at the vascular smooth muscle GLP-1 receptor, and in normal mice with those intact receptors, we see very nice reduction in blood pressure and increase in urine water and solute excretion. We see the same thing in diabetic mice. We have preserved actions of semaglutide to lower blood pressure and to increase urine water and sodium excretion. But if we knock out the vascular smooth muscle GLP-1 receptor, we lose the blood pressure lowering effect, and we clearly attenuate the effect on urine flow and urine electrolyte excretion. But if we then circle back to type 2 Cre mice that have no GLP-1 receptors either in their endothelial cells or in their immune cells, we still see blood pressure lowering. So at least from our perspective, and this is unpublished data, we think there's a role for the vascular smooth muscle cell and the GLP-1 receptor in that cell in the blood pressure lowering that we see with GLP-1 medicines. So let's circle back to another exciting area, which is peripheral artery disease. In people with this disease that have severe disease in the lower extremities can't walk very far without having pain. We call this intermittent claudication, and it really limits walking distance and quality of life. Well, there was a trial, the STRIDE trial, that administered semaglutide to these people for a year, and there was an improvement in their functional ability to walk without pain. So how does this happen? Does GLP-1 reduce atherosclerosis? Some people think so. How might that happen? Well, again, we can model this in mice. We can say, where are the GLP-1 receptors in the mouse aorta? And most of them are in endothelial cells. There are very few receptors in immune cells or vascular smooth muscle cells. When we knock out these receptors and induce atherosclerosis and treat the mice with semaglutide, we still see a reduction in atherosclerosis, even though we've removed 95% of the GLP-1 receptors from the mouse aorta. So how does GLP-1 reduce atherosclerosis? It's probably through other cell types that ultimately converge to produce this plaque. Now, a very exciting area of GLP-1 action relates to inflammation. And when you think about the location of the L cells that make GLP-1, these are not located in the duodenum or jejunum where you would think an incretin might be, like GIP. They're located in the distal gut. What's in the distal gut? Aside from some digested food products, lots of microbiota, lots of viruses, lots of fungi, and their byproducts, both structural cell wall products as well as metabolites, are very good ligands for activating GLP-1 secretion from the L cell, which locally attenuates T cell driven inflammation. But does this explain all of the anti-inflammatory effects of GLP-1 that we see in people? I don't think it does. Now, how do we know that these anti-inflammatory effects are weight loss independent? Well, we have both animal and human data showing that one injection of GLP-1 reduces inflammation. So there's no weight loss with one injection. We also have clinical trial data, as I'm showing you here. This is a trial in people with type 2 diabetes who either got the SGLT2 inhibitor, empagliflozin, or they received once daily oral semaglutide. And let's just look here at the body weight difference between people who got semaglutide and people who got empagliflozin. You can see it's a few hundred grams at 26 weeks. It's not very big. But look at the difference in the reduction of CRP. It's very small with empagliflozin, but quite substantial with oral semaglutide. So it's, again, clearly not weight loss that's driving these anti-inflammatory effects. Now, where are the GLP-1 receptors in the immune system? There's not very many of them. They're predominantly localized to these gut intraepithelial lymphocytes, these T cells in the small bowel and some in the large bowel. And then there are a few in other immune cell types, in NK T cells as well as in exhausted T cells. So what we and others are trying to do is figure out what contribution to the anti-inflammatory effect of GLP-1 is made by these various immune cell populations that express the GLP-1 receptor. So we thought we would look at graft versus host disease to try and understand how important those T cells are in the gut that express the GLP-1 receptors. You know that in humans or animals, when we introduce an allogeneic hematopoietic cell transplant, we see a risk for graft versus host disease. T cells attack the gut mucosa, they attack the skin, sometimes the lung, and people can get very sick. And there's lots of GLP-1 receptors on those T cells that are attacking the gut. But we did a lot of transplantation studies and treated animals with semaglutide, or we used donor cells that did or did not have the GLP-1 receptor. And surprisingly, we don't see any role for the GLP-1 receptor in those T cells. And there are lots of them there in the pathophysiology of gut injury in graft versus host disease. Well, what about the microbiota? They influence so many aspects of physiology, whether it's locally in the gut, it could be in the brain and the gut-brain axis, could be in other organs, the liver especially. Do we think that the type of gut microbiota we have influences some of the outcomes we see with GLP-1 medicines? And so far, we don't have very much data to support a role for microbiota in the actions of GLP-1 medicines. Here I'm showing you the microbiota profiles from those graft versus host disease experiments that we did with semaglutide. And yes, semaglutide does change some of the proportions of the species of microbiota, but they don't appear to meaningfully contribute to the intestinal damage and the lack of effect of semaglutide in this model. So what about the brain? There are GLP-1 receptors in the brain that transduce an anti-inflammatory signal that's meaningful systemically. In other words, if I induce inflammation, either with lipopolysaccharide or with a cecal slurry, a polymicrobial sepsis, so this is systemic inflammation, and then I treat with a GLP-1 medicine, I can reduce that systemic inflammation. But if I block subsets of GLP-1 receptors in the brain, either pharmacologically or genetically, I no longer have the same extent of attenuation of inflammation in the periphery, for example, in white blood cells or in the lung. So we're trying to figure out how important is the brain GLP-1 receptor system for some of these systemic anti-inflammatory effects that we see with GLP-1 medicines. And I think the answer is going to be it's important in some instances, but it's not the whole answer and not always explaining the anti-inflammatory effects and benefits that we see with GLP-1. And as we get the new drugs, they're all going to have different effects on inflammation. So here we've induced inflammation using 3 different ligands that activate toll-like receptors. And when we give exendin 4, you can see that we reduce systemic levels of TNF alpha. If we then do that experiment where we've knocked out the brain GLP-1 receptor in neurons, we no longer reduce TNF alpha in the systemic circulation. But what about if we give tirzepatide? We still see a reduction in TNF alpha in the systemic circulation, even though there's no brain neuronal GLP-1 receptors. So what this reminds us is that every time we tinker with GLP-1 medicines, and you're going to have some elegant lectures at this meeting that discuss the new emerging combinations, we have to remind ourselves that we have to go back and revisit our concepts of how these new medicines might work in multiple different domains. So what about metabolic liver disease? We had the results of the Essence trial. This is a phase 3 trial with semaglutide once weekly in people with metabolic liver disease, and there was a benefit. There was resolution of inflammation and no worsening of fibrosis in people with MASH. And this medicine was now approved very recently in the United States for the treatment of MASH. So how does this work? It's very logical to assume, well, this must reflect the weight loss that we see in this circumstance, because driving fat out of the liver happens when we lose weight and will be associated with benefits such as reduced inflammation and reduced fibrosis. But several years ago, we knocked out the GLP-1 receptors from immune cells and T cells in the liver. And despite pretty good weight loss that's preserved because the receptors are there in the brain, we saw a reduction in the benefit of medicines like semaglutide in mouse models with metabolic liver disease. So implying that there are weight loss independent benefits, perhaps mediated through GLP-1 receptors on rare cell types. So that's mice. What about humans? And we've started to get some of the post hoc analysis of the liver trials that Novo Nordisk has done. And what you can see here is a presentation from Professor Newsom, where he's plotted the importance of weight loss dependent versus independent actions on key targets such as reduction in liver enzymes, fibrosis, stiffness, etc. And remarkably, you can see that a lot of the benefits for many of these different pharmacodynamic endpoints are substantially weight loss independent, just like we see in mice. So let's take a look at another way to look at this, which is to tease apart these actions in mice with metabolic liver disease. So we can induce metabolic liver disease, give semaglutide, the liver gets better, there's lots of weight loss. But then we can knock out the receptor from the brain, so there's no weight loss at all, induce metabolic liver disease, but semaglutide still makes the liver better. And then we can say, let's go back to those endothelial and immune cells in the liver. If we knock those out, now semaglutide no longer improves liver health despite tremendous weight loss in the mice. And we can introduce this adenovirus to inactivate these receptors only in the liver, and we lose the hepatic benefits of semaglutide. So obviously weight loss must be important and must contribute to some of the benefits, but it's really not the whole story. So among the biggest mysteries and areas of excitement is the brain and neurodegenerative disease. We've had 5 trials in Parkinson's disease, 3 looked promising, 2 not so much, all with exenatide-based medicines. And we have all this preclinical data and real-world data that shows the possibility that GLP-1 might benefit this area. But 2 EVOKE trials with semaglutide, oral semaglutide, will report out hopefully this year, and we don't know what the results will show, but a very important set of trials. Now, we've tried to tease out these mechanisms in animals, and we definitely can show in some experiments a reduction in neuroinflammation when we give GLP-1 medicines. But here we use 2 different widely used models of neurodegeneration that mirror familial Alzheimer's disease. 6 months old, 12 months old, male, female, semaglutide, tirzepatide. The drugs work, glucose is reduced, body weight is reduced, but we don't see a benefit. We don't see reduced plaque intensity. We don't see reduced astrocyte activation. We don't see reduced glial cell activation. We don't see reduced levels of neuroinflammatory biomarkers in the brain. So, I'm hoping that these mice will not reflect the results that we see in humans, because I would love for the EVOKE trials to be positive, but it's been difficult for us at least to model out the potential mechanisms. And as we celebrate, more than 20 years of GLP-1 in the clinic, we also need to celebrate the innovation that will bring us the next chapter. So the fantastic actions of adding on different bits and pieces, whether it's GIP and glucagon and amylin and other epitopes, to make these medicines even more powerful. These are experiments that have been, really pioneered substantially by a terrific partnership with Professor Richard DeMarchi and Matthias Tschöp and their colleagues, really illustrating to us the power of new combinations that rival the benefits of weight loss that we see in bariatric surgery. So much more excitement to come and more science needed to understand the risks and benefits of these medicines in the clinic. So those are the remarks that I wanted to share with you today. You can see that we've made some progress in understanding the mechanisms, but some of them still remain elusive and more work needs to be done. I hope everyone is having an excellent conference in Korea, and thank you very much for inviting me to share some of our work and our understanding of the field with you. Thank you.
Okay, thank you, Professor Drucker. So we want to move on. Next speaker. The second speaker is Professor Matthias Tschöp from Germany. So Matthias, can you hear me?
Yes, I can hear you. Hello.
Okay, so let me introduce you briefly and then you can start your talk, okay? So the Professor Tschöp is now CEO of Helmholtz Munich and the professor in TUM School of Medicine. I mean, I can say he's a pioneer in development of multi-agonist hormones such as GLP-1 and GIP-1, dual agonist. And then he is one of a person who redefined our field in obesity and metabolic disease treatment. So today he will give a lecture about the discovery of multi-receptor drugs, the end of the obesity and type 2 diabetes. So please welcome him with big hand. Matthias, you can start your talk from now.
All right. Well, thank you for the kind introduction. Two disclosures. One, I have a little virus infection, so my voice is not what it used to be, but I hope you can hear me anyway. And also, I'm not working at Helmholtz or TUM anymore. I changed jobs. As of this month, I'm president of Ludwig Maximilians University in Munich. The research stays the same though, and I'm delighted to get started and follow the previous fantastic talk by Daniel Drucker. So my talk is supposed to be about the discovery of multi-receptor drugs and if that could have something to do with the end of obesity and type 2 diabetes. Let me see. Yeah. Well, all of you have seen that there's a lot of dynamic in the field now and a lot of excitement with new drugs that are giving substantial benefits in terms of weight loss. And they seem to do things beyond that. Since The Economist titled a while ago, actually a year ago, the everything drugs, isn't this just for obesity? Is there more? Can we affect addiction, Alzheimer's and other things? Now, it's really relatively simply coming down to just a few compounds, a few ideas. The single agonists, and Daniel Drucker has just been reviewing a lot of their work and their benefits, and several versions of dual and triple co-agonists, single molecules that have the capability of addressing one or the other or a third receptor, and in concert activate all three. Now, this isn't something that came about overnight. All of these drugs are the result of a large journey for more than 100 years. Folks have been trying to understand gut hormones and how they contribute to metabolic control and, in the end, also to satiety control in the brain. I started in 1993 in this university hospital with that question in mind, and just refused to believe that this obesity pandemic that we are already seeing back then was something that we just never could stop. Now, obviously, obesity decreases life expectancy, and there are related complications that are massive. And if you want to cut it short, there are millions of lives lost with obesity every year. That seemed sufficient of a motivation to go into that field. And it wasn't for the lack of trying. There was a pipeline, so to speak, globally with obesity drugs, but either those weren't really going beyond a placebo effect, or they had significant toxic effects that precluded them from getting approved or staying approved in the US or in Europe. Now, our mission was to, go out there and try to find new drugs that would decrease body weight by 20 or 30%, do something that, we only know from a bariatric surgery treatment, gastric bypass to function. The first thing that really impressed me was the discovery of leptin by Jeff Friedman, and it really pushed me into science away from clinical work, because I found it so fascinating that there's a new hormone that for the first time really reliably regulates body weight, the whole genetics behind it, and that it works in the brain. So when, as a postdoc, we observed that there's another hormone that regulates food intake and body weight, which is counteracting leptin, then we were even more excited, even though there were two disappointments. Leptin alone wasn't working to treat obesity sufficiently for a number of reasons, and ghrelin antagonism wasn't either. So it seemed to be more complicated. Well, we learned though that when you look at these two hormones, they act in the brain, and when you look at the brain also seems to be where obesity happens. When you look at the entirety of genetic studies, and Sadaf, Giles, and Steve really are representing that genetics crowd that has figured that out. So we sat down together, Richard DeMarchi and I. Richard, a fantastic peptide chemist. We met at Eli Lilly and then both went back into academia and said, well, can a peptide chemist and a physiologist, ex-physician together come up with ways to make new drugs that are doing what we need them to do, up to 30% weight loss, being safe, and working well in humans. And our principal guiding strategy was to say, well, we want multiple receptors to be addressed at the same time, because it seems that Mother Nature has put it in place in a way that numerous signals in a pattern that affect what's happening in the brain sufficiently so that there is a palpable effect, a sustainable effect that we want to see. And we thought doing that with exogenous molecules it seems tricky, and we know that from CB1 receptor antagonists, etc., that usually comes with side effects. But what if we use Mother Nature's toolkit and use gut-brain signals, which are in place already, to find the right receptors in the brain, in the few right neurons where they affect only appetite, satiety, and body weight change. Okay, so, but where to start? At that time, then, there was an explosion of signals that were accused of playing a role, and we really focused in quickly on the glucagon family. Glucagon being a hormone that, according to textbook, is doing one thing: it increases blood glucose, but it does other things as well. It responds to nutrients, and it can increase lipolysis, increase satiation, decrease food intake, increase thermogenesis, increase energy expenditure, so help with the burning of calories. Another thing was interesting for Richard specifically. He looked at the family of peptides and said, well, there are a number of sequences that seem to offer us engineering of one hormone, so that the result looks like a gut hormone, maybe won't be rejected and attacked by our own immune system, but actually affects two receptors or three receptors. So a superhormone seemed to be possible based on the chemical structures. Well, you first got to start making compounds to work with, and making a glucagon receptor agonist isn't that trivial, because you want longer half-life. These hormones have a very short half-life, and you can't really do pharmacology with them. They need to stay soluble, and they need to have a strong effect at the receptor. Lots of small steps with chemists around the world contributing have enabled that line of research. Here you see the Heloderma horridum. John Eng was the first to find out there's a version, the saliva of this animal, that can be used for longer acting extending GLP-1 to be isolated. But that has a particular tail, and this tail then, we could learn, you can put on other hormones as well and make them longer acting and still have them soluble. So that was done, and this was one of our eureka moments where we saw for the first time in 2006 that a longer acting glucagon molecule can decrease body weight significantly, as much as a long-acting strong GLP-1 receptor agonist. Showing us, well, glucagon has that potential to add to weight loss. But what if you combine the two? Everybody we talked to said at the time, you're going the wrong way, that's ridiculous. You can't have a glucagon receptor agonist and think that helps for patients with prediabetes and obesity, because every pharma company in the world is looking for an antagonist, and he will drive blood glucose up. So we went after that anyway, and Richard made a dual agonist, a single molecule with GLP and glucagon. Now, that goes back to the discovery of GLP bioactivity, something that Svetlana Moisov and Joel Habener were maybe the driving force, but Daniel Drucker and Jens Holst also made significant contributions to that discovery, showing that there's a bioactive component of GLP-1 that can trigger insulin secretion. Nobody was talking about body weight yet. Actually, later there were then observations that body weight could be affected, and the first clinical study showing that a GLP-1 could affect human obesity was led by Richard DiMarchi and Suad Efendic. Both had a patent on that, and they could show that this is doing something to body weight, not only to blood glucose. The first observation that body weight can be affected was done in rats, by the way, by Steve Bloom, who gave GLP-1 compounds intracerebral ventricular. Oh, back to our studies, though. So knowing from these studies, from these colleagues, that GLP-1 could have satiety effect, could affect body weight, and from our own studies, the glucagon receptor activation could do that too. Dual agonists were made, single molecules, not two molecules glued together, but one molecule engineered to be able to activate one or the other receptor. And indeed, there was body weight that could be driven down significantly, 20–30% in a short time. That came without problems with insulin levels. They improved, and it improved glucose tolerance. So the GLP effects seem to override the bad glucagon effects on diabetes. This was published as the first dual agonist, the first polyagonist being superior to monoagonism in the treatment of obesity and diabetes. By now we know there are clinical data. Here's some out of Boehringer Ingelheim showing that they can do exactly that, decrease body weight safely in humans with survodutide, a glucagon dual agonist. I think we're going to see more data in the next year from this and other companies on that. But we wanted to do more. We said, well, there's another one of these hormones called GIP, and GIP seemed interesting to us. It wouldn't have the diabetogenic liability of glucagon, but it would indeed maybe also help not only with fighting diabetes, but with fighting body weight. So we made long-acting GIP, and I think that's a very important point, that long-acting GIP, like longer-acting GLP, do a little bit different things, maybe hit different receptor populations than just a very short-term natural hormone. And you gotta go make them, test them, see which ones are working at the mouse, rat, human receptor. You test if they are affecting blood sugar level, which they should. And then you look at the body weight effect. And interestingly, GIP receptor agonists that are a little longer acting decrease body weight. And they do that only if there's a GIP receptor around, so they're specific. Again, everybody said, you're crazy, wrong way, stop, you're embarrassing yourself, because you need a GIP receptor antagonist. GIP is a bad guy, so you need to stop GIP, and we, stubborn as we are, disagreed and said, no, we see great effects with GIP receptor agonism, and we want to pursue this, also having in mind we want dual agonism. And dual agonism is so much easier to construct when you have two agonists, rather one antagonist, one agonist, where you then have massively different action to be balanced. Now, there are first data out of Lilly, for example, showing, I think, new data just emerging now, that a GIP receptor monoagonist that Lilly has made can drive down body weight in humans. So indeed, GIP receptor agonism seems to be the right thing to do. All right, so with the first dual agonist, a dual incretin, a GIP receptor, GLP-1 receptor agonist, we could show for the first time you can dose-dependently drive down body weight. And we published this shortly thereafter in 2013, 12 years ago. And later, we're part of the first clinical validation study showing that indeed you improve blood glucose, you improve body weight with these these dual agonists. This was collaboration with Roche and Novo Nordisk, showing also that this is, post-hoc analysis-wise, better than monoagonism. There's a very short phase 1b study in 12 weeks. Today, we know that that concept is really fantastic and so superior because Mounjaro or Zepbound is such a GLP coagonist, more refined, different sort of acylation on the molecule, and a different ratio. Ours was pretty balanced, GLP, and Mounjaro is heavily balanced toward GIP. But the exciting thing is this principal class of drugs that we had first discovered is capable of decreasing body weight by 30%, 23% on average. It shouldn't be overlooked that this is also tirzepatide. Mounjaro is doing great things for diabetes therapy. When you look at the right side, 50% almost have a normalized blood glucose. How does that work? Well, we think one point where the dual agonist is superior to monoagonism has to do with the side effect profile. They all have sort of a prevalence of side effects that are first look comparable, but relative to the benefits of tirzepatide, which gives you more like 23% weight loss compared to 15 or 17% weight loss with a monoagonist, there's less side effects. With this model here, the Suncus marinus musk shrew, Matthias has nicely shown that GLP-1 triggers nausea and vomiting, and GIP doesn't, and the combination doesn't. So GIP seems to counteract even the toxic effects of GLP-1. Similar might be true for gastric emptying, which is more of a problem in the monoagonists and not the case with dual agonists. Question is, which cells are mediating this? Timo Müller, who took over my lab a while ago as my successor, has figured out, well, you can remove GIP receptors from the brain and then you lose the additional benefits that GIP gives you. Then he has been looking for which cells these are and identified that GIP decreases body weight via GABAergic GIPR neurons, and knocked the GIP receptor out there to prove that. Now, we wanted to understand better why there is still discussion that GIP receptor antagonism could also be beneficial. There's a company by the name of Amgen that's pursuing that, while we are clearly seeing that agonism seems to do the job. So we went with my grad student, Roald Gutgesell, into these studies and compared and said, well, is it same? Is it different mechanisms? So the first thing we observed was that there is preserved weight loss in VJET GIPR knockout mice when you give antagonism. That means the agonism that's mediated through these GABAergic neurons is not mediated, also mediating antagonism. So different mechanisms, different neurons with GIP receptor are mediating the benefits of agonism and the weight loss benefits of an antagonism. All right, so same goes for peripheral neurons, where you don't see that being blocked either. And what's interesting is that you can knock out the GIP receptor, and then you don't have GIP receptor antagonism-mediated body weight effects, fair enough. But you can also knock out the GLP receptor and see, I don't have this data here, it looks like, And you also, oh, here, yeah, you see the GIP receptor antagonist given to GLP-1 receptor mice that are haven't knocked out GLP-1 receptor. And you lose the effects of the GIP receptor antagonist. So that's interesting. The GLP receptor is really needed so that the GIP receptor antagonist can do anything. All right, well, we went through these RNA sequencing studies in the hindbrain and the hypothalamus, very complicated, very refined, very expensive, and saw that there's clearly different patterns. The patterns of GIP receptor agonism and dual agonism look very different than the GIP receptor antagonism patterns, which look so much more like GLP-1 receptor agonism. Those look to be more similar. Again, in the hindbrain, GIP receptor antagonism seems to mimic GLP-1 receptor agonism. So there's something much closer between blocking the GIP receptor and activating the GLP-1 receptor. While on the other hand, GIP receptor agonism is more similar with the dual agonism. All right. Much about these very complicated pictures. So we, from these results, would have predicted that GIP receptor antagonism should enhance GLP-1 receptor agonism and therefore give you more side effects, while GIP agonism should counter the side effects of GLP-1 receptor agonism, which you see here. Now, there's a study out just, I don't know, a few days ago in the New England Journal, and there the side effects of maridebart cafraglutide, this compound by Amgen, are examined. This is a dual GLP receptor antagonist and GLP-1 receptor agonist. And to cut a long story short, there are unprecedented levels of nausea and vomiting, which was what we would have predicted. 70 to 90% and up to 90% of vomiting. If you imagine you're a patient, and this is a once a month drug, you're being injected and then you vomit for a month and can't stop. I'm not so sure that is what any patient would desire. All right, back to our dual and triple agonists. We then made a triple agonist on glucagon, GLP, and GIP. This took us a long time to figure out the right balance here. We found one that worked. Again, this molecule just looks like a normal gut hormone and can now activate 3 receptors. And with that, we found something that's twice as good as the dual agonist. So this is something like tirzepatide here, and this one is a triple agonist that works twice as well. Fat mass decreased, cholesterol decreased, blood sugar decreased, insulin levels decreased, liver got completely cleaned of fat. This was also published 10 years ago. And again, pharmaceutical industry picked that up and made a number of versions of that. This is the Lilly version, retatrutide. We're all looking forward to see the phase 3 data. Here is phase 2 data showing in a couple of weeks, 48 weeks, 24% weight loss on average at the highest dose. We really wanted to know then, is this for real, or are we doing something else coincidentally? But with a triple receptor knockout mouse, a mouse that doesn't have GIP, GLP, or glucagon receptor, we could show that you lose all the effects of the triple agonist, meaning it's not doing anything beyond activating these 3 receptors. All right, so there is now actually more than 20 next-generation therapeutics for obesity and diabetes, with many pharma companies, many in phase 2, phase 3. With these drugs, going from monoagonists that got even better to dual agonists to triple agonists, I think we have closed the gap, or we are about to close the gap with maybe retatrutide. One is expecting 28% and more weight loss to what bariatric surgery can do. I summarized the effort that the discovery of dual and triple agonists delivers safe obesity therapeutics of unprecedented efficacy, and the benefits approaching those of gastric bypass. Well, we still think there's more to do. We still think there are patients that require even better, even more efficacious obesity drugs. And the reduced comorbidities, when you go from BMI 40 to BMI 27, 29, it's amazing how many comorbidities are left, right? So maybe we need drugs that are focusing, in addition, more on comorbidities than simply on body weight loss. And this is something that we are trying to address with our new startup called Blue Water Biosciences, where we're also revisiting another approach that we discovered earlier, where we have steroid hormones, small molecules on peptides, and deliver them specifically to peptide receptor carrying cells, sort of precision metabolic medicine, and we're hoping to put those in the clinic shortly to offer additional versions of this. So with that, I'll end and say metabolic precision medicine, it's not the beginning of the end, but the end of the beginning of obesity. All of these talented individuals have worked on these experiments, and you can tell that I'm a significant fan of dual, triple, quadruple, quintuple agonism rather than monoagonism. Thank you for your attention.
Okay, thank you, Professor Matthias. Thank you for your great talk. So we don't have any Q&A, so maybe we can meet another conference. Thank you. So we will move on next speaker. The third speaker is Professor Gerhart-Hines from Copenhagen, University of Copenhagen. He's work on the Novo Nordisk Foundation Center for Beijing Metabolic Research. His research is focused on the field of muscle and adipose tissue and integrate circadian and metabolic signal to maintain energy balance. So today he's going to talk about after the incretin revolution, exploring the next horizon in cardiometabolic disease pharmacotherapy. Jack, please.
Thank you very much. And yeah, thanks for the opportunity to discuss some of our recent efforts and others. It's never easy following up two extraordinary figures in the field, Dan and Matthias, talk about walking in the footsteps of giants. But I will do my best. So I think really nicely picking up from where Matthias left off, I think we'll start with, I think this is one of the last slides he presented, and that is really thinking about obesity in the context of the current therapeutics versus the previous gold standard, which was, of course, invasive bariatric surgery. And I think you can appreciate that in people living with only obesity and no other existing comorbidities, that we are now approaching near surgical correction with the different pharmaceuticals. But I think the important consideration here is when you look at individuals, the 450 million living with both obesity and type 2 diabetes, this picture looks quite different. We could see it in one of the earlier slides from Dan when he was showing the different step trials. There was one where the weight loss was only about 6-7% over placebo, after placebo, and that was in individuals living with type 2 diabetes and obesity. And so here, I think we can appreciate that the innovation bar is actually there's still quite a need, and this represents several hundred million people. Oh, there we go. That might be better. Then the other consideration, I think, when considering the next generations of things, is related to weight maintenance. So, of course, we know that we know from the Swedish obesity trial that bariatric surgery is able to sustain weight loss by and large over several decades. The data still remain to be determined whether the pharmacological interventions are able to provide this degree of sustained activity. So for the sake of time, I won't belabor this too much, but this is just this is a slide that a very talented PhD student in my group made, where she, on the left side, took the different molecules that are currently targeting for type 2 diabetes and pinpointed the organs, the primary organs of action, and then did the same for obesity. And you can see, of course, some of the same molecules exist on both sides, but they're maybe exerting their actions through different organs. And I think for obesity, the real key is that you can see that it's not a accident that the brain, or at least neuronal signaling plays a huge role versus the remission of type 2 diabetes. I would also say that this still, I think, leaves a lot of opportunity for the periphery now that we have a lot of agents that are targeted in the CNS for appetite suppression. So I think this shows that, yes, obesity is a brain disease, but I think now there's other add-on possibilities throughout the body that we can target. We also live in what I consider to be sort of the golden age of obesity pharmacology, and we're in this sort of constant battle/discussion that we're watching between the different pharma companies over, now we have injectable peptides is what we start with, then we have oral peptides, we have oral small molecules, now with antibodies like from Amgen that Matthias was discussing. So what will sort of win out? Soon we'll have once monthly, perhaps even longer injectables. Is this more appealing to patients as opposed to taking a daily pill? It will remain to be seen. But I think the next several decades is going to see a lot of amazing innovations. Now, when I look specifically, at least in our group, and I think if this was touched on by both Dan and Matthias, where are the unmet needs? Where do we need to be focusing on in the next sort of post-incretin and incretin era? And for our group and our focus, really being able to increase metabolic rate and energy expenditure has been a passion for ours for about a decade now. And you could see this in the slides that they presented previously on the different drug development. So Matthias showed a drug from the 1930s, one of the first approved obesity drugs. This is actually the uncouplers, the mitochondrial uncouplers, dinitrophenol, which was very efficacious in causing weight loss right up until you died of cardiovascular failure. And so I think that's why I also put safe and energy expenditure have to be paired, but this is still a sort of a holy grail that remains to be unlocked. We also heard from the recent data on the maritide molecule from Amgen that tolerability, not just with that molecule, but with the whole incretin class remains still a problem. Talking to clinicians or patient advocacy groups, many of them say if you could deliver even less weight loss, but have it be completely nausea-free, this would still represent blockbuster capacity. So I think that even though the tolerability has increased as the efficacy has improved, so people are willing to tolerate it more now that they can see a lot of weight loss, it's still something that is a concern. The other thing, and I mentioned this with the weight maintenance on the previous slide, so the ability to avoid this pervasive weight regain. So a lot of times, I think after 2 years, for instance, 60% of people are no longer taking their GLP-1 at the diabetes dose, and it's even worse for the obesity dose. They regain the weight, they're regaining it mostly as fat, and in some ways are putting themselves in a worse position. So this is really a major pressing need. I already commented on the variable response among type 2 diabetic patients, which again, I still find to be quite flabbergasting. The other thing, this is something that I'm not sure that biologists like myself can help address. So the short stay time that someone's actually on the medications, this is probably due to cost, reimbursements. That's would require a whole other conference to discuss. But again, it's something that I think is we just need to keep on the backs of our minds. This is probably more a chemistry problem. I've discussed this a lot with students, also the pharmacologists, and that is you can make the world's greatest innovation, and we're seeing this now with triple and soon to be quintuple, quadruple agonists. But if you can't synthesize it for the masses and provide it to the people who need it, then it's less of a widespread impacting innovation. And then finally, I think something that's come in the news, at least in Europe and the US quite a bit, is this issue with lean mass / skeletal muscle. How much of the loss in this is due to physiological loss in body weight? How much is actually something detrimental? This is, I think it remains to still be fully determined, but I think that when we talk about future drugs, if you can have something where you only lower fat mass and completely preserve your muscle, I think this would still be desirable over, especially in the elderly population, where even 10, 15, 20% loss in muscle might be detrimental to health. So this is sort of the playing field that we work in. And again, I think still this holy grail is what guides us. So we've previously made some inroads into this by identifying different cell surface receptors that we could potentially use as either add-ons to incretins or help to bolster metabolic rate. This is just work from a couple years ago where we found one particular GPCR, GPR3, and just to really go through, because again, it's already published, but I think to highlight some of the key data that's related to obesity therapeutics. In a mouse model, preclinical mouse model of obesity, where we genetically activate GPR3 only in fat cells, if you put an animal in a metabolic chamber and be able to measure its oxygen consumption, so you're measuring how much calories it's burning over several days, you can see the natural oscillation of energy expenditure that we see during the day. When we turn on GPR3 with injections of tamoxifen, I think in red, which is where we have the overexpression, you can really clearly see that there's an increase in energy expenditure in these animals. Now, obviously, this starts lowering weight as it already at that point, but even prior to weight correction, when the animals would still be considered obese, their glucose control is that of a lean mouse. So we see that an obese animal has a really high, they can't undergo glucose excursion and remove the glucose from blood, whereas GPR3 in the adipocytes completely clears this. This is because that is essentially the fat cells turn into a big sink for macronutrients, and this is something that I think is an appealing therapeutic concept. And so here you can see across different tissues in the body of the animal, it's really the brown adipose that's taking up, in this case, lipids. We see the same with glucose. So it's essentially this macronutrient sponge. And then this is a different animal model, because this is one where we've constitutively, so chronically activated GPR3 in the fat cells. It renders animals completely resistant to obesity. So these animals in blue are eating the same as their litter mates in gray, but they're simply 15, 20 grams difference in body weight. So this is something that we're still pursuing, but this is probably more of a small molecule type angle. The GPR3 represents difficulty in drug ability for other reasons, and so that's why what I'll focus a lot on today and what we think is the most promising from our standpoint is the neurokinin 2 receptor. So this is the team in my group working on this. This is something that we published just late last year. It's also the source foundation for my biotech company, Embark Biotech. And essentially how this works is we've developed long-acting neurokinin 2 receptor agonists. They act through two different means. The first is they act on areas of the brain. We know at least the hindbrain is involved. We think other areas as well. This directly causes an appetite suppression that we've found to be non-aversive. And in non-human primates, so rhesus macaques, it resulted in no cases of nausea at all compared to GLP-1s. And then in parallel, that brain signaling, as well as direct signaling in the periphery of these agonists, hit a number of target organs, including the fat, but also the gut, muscle, pancreas, and liver. And this serves to increase metabolic rate. We see, and I'll show in a couple slides, we see a pretty substantial increase in energy expenditure. And this also leads to an increase in peripheral insulin sensitization. Now, calling back to some of the unmet needs I mentioned on the first slide, I think what we found very interesting was that even in the context of non-human primates that were almost type 1 diabetic, so they were so diabetic on diet and had undergone so much pathology with their pancreas that they were actually hypoinsulinic. These animals actually responded the best in weight loss to the neurokinin agonist. So this would present almost, if anything, an opposite regulation compared to the GLP-1s. And this is just, and then of course, I think all the cardiometabolic diseases that I've mentioned would be a benefit. And so the slides of unmet needs I mentioned in the first place, again, we don't have the synthesis and scale up, but a lot of the other ones, we think this could actually be a very nice add-on to. So a brief primer, since we've heard from Dan, a beautiful introduction about the GLP-1s, and then from Matthias, GIP, and glucagon. This is just a bit of a primer for the NK2 receptor if you're not as familiar with it as I am. So this is a family of 3 receptors. They're again all of the same general GPCR class that the GLP-1, GIP, and glucagon are. So obviously a very druggable class. But I think what is really notable to consider is that this class of peptides, the tachykinins, the ligand for the first one, substance P, is actually called the pioneering peptide. It was in fact the first neuropeptide cloned. So it's of quite significance. And this family, this crisscrossing pattern that you see is not by accident or not to confuse you. It's because this family is a highly promiscuous family of neuropeptides. So fortunately or unfortunately, a lot of the endogenous ligands can easily cross-react and activate the other receptor. Now for drug development, this poses a big problem because as you can see here, we are focused on neurokinin 2 receptor, and you can see the pharmacological curve with NKA, neurokinin A. It, of course, beautifully activates its own receptor, as we would anticipate, but you can see it also activates almost to the same degree as the other endogenous ligands, NK1 and NK3 receptor. Now, as I mentioned, that becomes a problem because neurokinin 1 receptor is linked to nearly every pathological problem you can imagine. So pharma companies have actually been trying to antagonize this because it was linked to anxiety in the brain, bronchoconstriction in the lungs, so a problem for asthmatics, increased heart rate, inflammation in the GI tract, you name it, NK1 probably causes it. And so that's been very problematic for the last 30 or 40 years since these classes have been discovered to try to do anything with NK2. And so this is where when our academic group came to NK2 and found it interesting based on some adipose biology, we realized that one of the first things we had to do was create a selective agonist. And this is where our first collaboration with Novo Nordisk came into play. So using the massive expertise and help of Novo Nordisk, we were able to work with peptide chemists and go from a molecule that, like GLP-1, has a half-life of 3 to 4 minutes in the blood, to something that's conducive with once weekly administration in humans, and something that is completely non-selective, as the black is now 1,000-fold selective away from NK1, and at least in animals, and we have whole body knockout animals, and it's completely selective. So as Matthias was showing at the very end with the triple agonist, those knockouts, that drug goes completely through those receptors. We've done the same for NK2. Now, because safety and selectivity is so important, I'll just briefly highlight, not to belabor too much on the molecular mechanism, but I think this is quite elegant work by a very talented student. So we were wondering how this selectivity might be achieved. So again, just to reiterate what I showed on the previous slide, you have NK2 that we have shown, and now others are showing, has a lot of beneficial effects. Again, insulin sensitization, calorie burning, appetite suppression, but the problem is this ligand is linked to a lot of other negative elements through NK1. And so we knew this from our first publication and through the work with Novo Nordisk, but we didn't know why this was. And you know, a lot of times you can't break through the barrier of making better drugs until you really know structurally what's driving this. And so this has been a fantastic collaboration with Shangyu Liu from Tsinghua University, where he actually made multiple cryo-EMs of NK2 with not only our selective agonist 1002, EB1002, but also the endogenous NKA and several other selective peptides. Now, not to belabor and go into the details of the structure and function, but I think what was fascinating was that we found that there are only several residues, in fact, 2 real key residues in the binding pocket of NK2 that are actually driving selectivity away from NK1. And if we, again, not to bore with sort of alphabet soup or the logistics of cryo-EM structure, but I think what you can appreciate is what Artem did was he took, if you start with the gray NK2, so this is the selective NK2 agonist. You can see that obviously the agonist is much more selective on NK2, so the gray line is far to the left. You can see in green, this is the signaling on NK1. This is the one we don't want to target. And you can see that it's obviously, this molecule 1002 is quite selective away, so it's about 1,000-fold selective away. But then in dark green, this is when he took NK1 and he simply mutated 2 residues in the binding pocket to make them like NK2 receptor. And suddenly, I think you can appreciate from the curve that it renders that molecule almost completely sensitive to the selective agonist. So this has a lot of implications for future drug development, and as we now start trying to make polyagonists, which again, I should say I fully agree with Matthias, I think the days of monoagonism are nearly over. Knowing how you can glue something together and what needs to be impacted is extremely important. And I'll use this slide also to just highlight another important point that I don't think came up in the first two talks. And that is the way this revolution was really facilitated was by being able to make GLP-1 long-acting. GLP-1 by itself does essentially nothing to body weight endogenously. But by putting a lipid on it and allowing it to sort of reside in the blood for up to a week, now we can see the substantial effect. Just putting that lipid on, which is supposed to make it long-acting, also impacts the binding to the GLP-1 receptor, and this NK2 is no different. So there's a lot, as Matthias was mentioning, when you start gluing things together, there's a lot of considerations that one has to have. So I think now that was just highlighting, I think, a little bit of the things that we consider from the really basic preclinical angle of how you would look at the current incretin therapeutics and what the bar is raised to move to the next level. But now just to show you again a few of the functions I was mentioning before. So some of the features of this sort of dual ability to control food intake and energy expenditure by one signaling molecule, which is previously not, that has not been has shown before. What we use as our normal routine or experimental setup is to take an obese animal and then inject our molecule in the context of metabolic chambers. So this allows us, if you're not someone who looks at this data regularly, every pharmaceutical company in the world is doing this, you can measure everything about the animal all the time. How it moves, what it eats, when it eats, and how it burns calories. And so the data look a little something like this. And I think, again, you don't need to know a lot about this setup to see that the black and white bars mean 24 hours. So this is looking at 5 or so days. And you can see that in gray, the normal animals are seeing a rhythm of energy expenditure, oxygen consumption. But then when we start injecting, indicated by the downward triangles, you can see we see a very nice and sustained level of increase in metabolic rate or energy expenditure. This is important, and I show this because if you inject adrenaline or one of the or dinitrophenol, the first obesity drug ever made in the 1930s, you'll see massive spikes of energy expenditure to the point where it will impact the animal's tolerance of the medication. So this is a very important point to see the tolerability of the energy expenditure. Now, in these same animals that are burning more, so again, you're burning more calories, which normally you would think would make you hungrier, and that is normally the homeostatic response. But in fact, what we see is under these same conditions, we see a pretty substantial reduction in food intake. So this combination, I think, really shows what I mean by neurokinin 2 biology being an exciting new paradigm where we not only reach a hypophagic or food intake reducing state, but also increase energy expenditure. And as you might expect, this, of course, is concurrent with a loss in body weight. I think I will skip this. So the other point that I mentioned and has come up in the talks is this idea of weight maintenance or weight regain. So when you stop a drug, we know from GLP-1s that you sort of bounce back quite immediately. So where could NK2 add promise to this area of the pharmacotherapies? So this is another study done by also a really talented neuroscience PhD student in the group, and she was just doing some dose finding studies with animals, trying different doses, and then normally we just sack the mice afterwards, but she was very clever and said, well, I'll just weigh the mice continuously even after I've stopped injecting, because she was starting other studies and thought, why not? She's in the mouse facility anyway. And I think as a parallel, I'll also show data we had from another study with semaglutide. So, as I mentioned before, we know that the GLP-1s, when you stop injections with GLP-1s, you see a bounce back almost immediately. So this is in animals, they begin gaining the next day. And what she found was over the same time frame, all the animals just simply plateaued even after we stopped giving NK2 agonism. So they maintained their weight. Now, unfortunately, we had to take them out of the metabolic cages, and our protocol is that we sack them, but we are repeating this study to see just how long that maintenance lasts. But I think this shows, again, an exciting dichotomy between the two and where the next future could hold. So then I think now I'll turn my attention largely to the brain signaling, because this is, I think, where we have the biggest source of mechanistic knowledge, and also, I think, the greatest support for translatability. So what you're seeing here, this is from our earlier paper, where you're looking at two composite mouse brains, where this signal is CFOS, which is just a marker of neuronal activation. And you can see that peripheral injection of an NK2 agonist pretty substantially increases activity within the brain. Now, I don't think there's a lot of brain penetrance. So Matthias mentioned the CB1 receptor antagonist with rimonabant and was associated with suicides and pulled from the market due to deep brain penetrance. This molecule, we think, is barely getting into the brain. It has the same lipidation as tirzepatide and as semaglutide. So this action that you're seeing is probably due to peripheral signaling back into the brain. And what we really focused on right now is the hindbrain. So this is and apologies to the neuroscientists in the group, this is a very oversimplified version within the context of a mouse. But essentially, the three areas I'll be talking about are the AP, the NTS, and the DMV, primarily the nucleus of the solitary tract and the area prostrema. And so, and then also for the purposes of some of the images I'll show, this is just if you were to take this region of the mouse brain, slice it down like this, and then turn it to face you, this is essentially what some of the histology you'll see. And this will come into play when we show some of the neuroactivation. So when we're looking at this setup, and we have, again, a vehicle versus a peripherally injected neurokinin agonist, again, I think you can appreciate the fact that there is a huge amount of neuronal activation in the nucleus of the solitary tract. Now, why should we care about this and why is this interesting? Every current drug, GLP-1, GIP, amylin, all target neurons within this population. So already we were kind of excited about the translatability of this molecule. But getting into this, understanding what the cell types are and why they might be relevant for human obesity pharmacotherapy is where we went next. So this was work done in collaboration with Jens Bruning at the Max Planck. And so what we did was we injected animals, we harvested the hindbrains, and Jens performed single nuclei sequencing, RNA sequencing on this region. And then we looked across known cell maps to see, okay, what are the populations that are being activated by neurokinin 2 receptor agonism. If you'll remember, Matthias actually showed something briefly from one of Timo Müller's papers on doing the same thing for GIP. And what we found was this glutamatergic population 3 was the one that was most robustly increased by neurokinin 2 agonism. But what is this GLU3 population? Why should you care? So before getting into the implications of this, I just need to quickly go through a high-level version of some of the program that Jenny used in the lab. So this is something that was developed by Tuna Pierce at the University of Copenhagen, and it's a program called SELECT. And what this does is it takes all the composite GWAS data, so all the human genetic data for a particular readout. In this case, we use body mass index for obesity. You can then take single nuclei RNA-seq of any particular tissue, in this case, again, it's the hindbrain, and you look across the different populations and see how the different genes are being expressed. And if those genes are obesity linked and they're highly expressed in a certain population, it gets a higher score, and so on and so forth. So the more obesity linked genes that are highly expressed, that population, as you can see in this example, and cell type B, gets a higher score. So that's just to preface the kind of data that we'll see. So now, and I apologize for a little bit of the blurriness, but again, it's actually not, you won't need it, you will be able to see this quite clearly, I have a feeling. So this is actually taking new unpublished data from Novo Nordisk and Martin Myers at the University of Michigan, looking at 260,000 nuclei in the hindbrain. So this is an unprecedented resolution of data set, and it goes even beyond the ones I originally was showing. And you can see above this line, these are all nuclei that when activated, they're significantly linked to obesity genes in the human population. And I think that, again, I apologize for the blurriness, but this is GLP-1 receptor. This is the CalCR, so targeted by amylin. So you're already in good company, that this is a nice validation that these drugs have already validated that targeting these neurons is a good thing. So we actually found that the GLU3 population activated by the NK2 are essentially as significant as these other known validated markers. So, but yet different. So a different population that has yet to be targeted. So we think not only is this strong evidence that targeting NK2 in humans would demonstrably impinge on body weight, but that also because it's separate neuronal populations, the potential for additivity or even potentiation of reducing body weight is even more significant. So I think when we're thinking about the future where polyagonism probably will be king, and where you need new modes of action, we think this is poised quite nicely. And this is just stating that. So I think to some of the final bits of data slides on this neurokinin 2 biology and why I think it has the capacity to be a real new revolutionary area is understanding again more about what these neurons are and why we should target them. I think for the sake of time, I will just jump to some more understanding of what could be signaling to these neurons in the NTS that I just showed you that are obesity linked. So there's two major ways that we think of. You can have signaling from within the hindbrain, so the area postrema can signal and project into the NTS because the AP is peripherally accessible through fenestrated capillaries. And we certainly see, the slide I just clicked through, we do see expression in the AP of NK2. But I think the real interesting thing that people often ask is what is co-localized, what is being expressed in those receptors as well. This is looking at a reporter animal where the green is containing the NK2 receptor, the red is containing, so in this case, GIP receptor. I think you can appreciate that there is a fair amount of overlap in this hindbrain region between GIP that we heard about from Matthias and NK2. This is just quantifying it. We see about 40% overlap. And again, since GIP, as you probably recall, was associated with less nausea, this matches with what we see, I think, with our lack of nausea from NK2 agonism. And then just to very quickly show that NK2 is not co-expressed with CalCR or amylin or GLP-1 receptor. So again, distinct and not in neurons that often cause nausea. I think for the sake of time, I'll skip through this part on the nodose ganglia. It's just other areas that we think we know are signaling in to these neurons. The final quick thing, because I think this is quite interesting and important, is understanding this element of nausea, as I've been discussing. So the data some of the data I've referred to when I say that this is a unique element is if you know what a condition taste aversion test is. So this is something that's also can be done in humans as well, but essentially you give an animal access to saccharin, sweet, it really loves it. You co-administer, in this case, we inject either GLP-1 or NK2 agonist, and then a couple of days later, you give them the sweet again and see, do they avoid it? Do they remember what it was like? If a drug either causes nausea or feelings of discomfort, it will avoid the next time you see it. And I think you can appreciate that obviously semaglutide, we see a pretty significant aversion. This is known. We see no aversion with the same dose of NK2 that can give the same food intake reduction. We don't see the aversion. And so, last data slide, I promise. So to test how the food intake is being regulated, and are there effects on macronutrients or sweet preference or other things that we can measure, we use the classic 2-bottle test. So this is, I won't show all these different macronutrients, but we essentially give either one pleasurable macronutrient or caloric macronutrient against water, and then we measure again the preference for these. And I think the data are pretty striking. So when we look at NK2 agonism using what is more of a hedonic, food desiring, pleasurable dose of sucrose, we can see a significant reduction in the sucrose preference, no change in water intake, and they're on the sucrose intake and water intake are on the same y-axis for a reason. You can see a profound decrease in sucrose intake over the course of, the bar is indicating the time of injection. That persists even after this. We see a pretty profound weight change even during that short time. And what's even crazier is the food intake. When you release the animals from injection of NK2, they become voraciously hungry. They eat almost twice the amount of food over the next period of days. This is usually very indicative of a physiology of energy expenditure. So we think we have something quite fascinating. And the very, very final thing is that if you know your neurobiology, sucrose at hedonic levels is often a sign of anhedonia or anxiety or other other problematic features for a drug. This is often evaluated using psychological behavioral tests like the open field test, where if you essentially have a video camera over a cage, and the more time an animal spends in the center, because mice don't like being in the center, the more time it spends in there, that's a normal, non-anxious mouse, whereas if it avoids it, then you can say that you have an anxious animal. So just to end on negative data, we absolutely see no difference in behavior of animals after NK2 agonism. So they spend the same amount of time in the center, so they're not flagged for anhedonia or anxious behavior. So just to wrap up, some of the elements I've shown you is that this neurokinin agonism represents a really fascinating biology where it both decreases appetite and increases calorie burning. It's the only single agonist to do that. The action in the brain appears to be significantly linked to BMI and obesity in humans. It appears to be reducing food intake without anxiety-like behavior. And finally, the repeated injections, we see this continued increase in energy expenditure, hopefully indicative of a more persistent response. And it was a huge team effort, and I look forward to the panel discussion later. So thank you very much.
Okay, thank you, Jack. It's very interesting talk. So we can do discussion next session together. Okay, so move on to next speaker is Professor Hyung Jin Choi from Seoul National University. So Hyung Jin Choi is a pioneer in the field of the neural circuit in the regulation of food intake and how GLP-1 regulate consummatory behavior. So related to that, he will talk about the GLP-1 and science of eating and how neuroscience is reshaping obesity treatment. Please, Professor Choi.
Yeah, thank you for the opportunity. Yeah. So today I'd like to share my thoughts and the data about the science of eating and the GLP-1. So thanks to the previous speaker that dealt with the neuroscience and the psychology of the animals' behavior. And I think this neuroscience and the psychology behind this phenomenon is reshaping our obesity treatment. And I think the GLP-1 is in the center of it. And I am an endocrinologist, used to treat patients. The reason I chose to move from the endocrinology clinic to the neuroscience field is because I saw that so many people die of the heart disease and stroke and diabetes. So another thing that I noticed was the rapid rise of this diabetes and this metabolic cardiovascular problems, and how our human race are evolutionarily becoming obese as we go on. And I think this phenomenon of the human race as a race becoming more obese, and it seems to be like a COVID pandemic that's happening over like 50 years. So during my times as a doctor, I saw people dying of cardiovascular disease, and we knew that is because of the diabetes and hypertension and dyslipidemia, because of the metabolism. But I thought that the center of all the problem was the eating problem. I think the eating problem is the major driver of the metabolic disease and the complication of the metabolic disease. So I thought I wanted to devote my whole life to find the fundamental cause of the driving force of this modern obesity pandemic. So, and I believe that I bet on the eating problem. So I think that could be the rapid rise of this metabolic and cardiovascular disease. And I thought that if we can find a drug that can that can treat this eating problem, then it can solve the metabolic disease and solve the cardiovascular disease. That was my belief 20 years ago. And I think GLP-1 and the Wegovy and the Mounjaro and all this therapy gave us a nice evidence that that is the truth, that if we can find a drug that can increase the eating behavior, then it can change the whole phenomenon. But my fundamental goal is to reverse this human race evolution to the obesity pandemic. So if we can find the psychological cause why do we eat so much, then we can reverse our this pandemic of the obesity, and maybe our grandchildren can have a better life. So that's why I wanted to devote my life on the science of eating, so that we can find a better cure for the science of eating, so our grandchildren can live a better life. So to do that, I chose the rodent model, mouse model, because it is so powerful tools to study the neural circuits. But and we want to continue on the primate and human samples and clinical trial, so we can have the whole picture of the science of eating. So during my slightly more than 10 years of doing the science of eating, I focused mainly on the desire of the eating, how our brain create this desire and motivation to eat, and the hunger, the need for the energy, and the palatability. So during this, my journey to the science of eating, I have been thinking about the whole picture of one cycle of eating. So this environment and the sensory, brain and motor, and the environment again. So it's a perception and behavior paradigm. So if your body senses the nutrients deficit, so if you lack your energy, then your brain, hypothalamus, calculate that you need like 500 kilocalories, and then your brain makes this motivation, desire, wanting to eat. And based on this motivation, if your brain decide to move to search for the food, as you find the visual or any olfactory cue of the food, then you approach to the food. And when you get near to the food, you have the whole picture of the olfactory touch and the taste of the food. And then when you put the food in your mouth, then you get this pleasure, the hedonic pleasure of the liking of the food. So after this pleasure of the food keeps you eating, and after several minutes, you become satiated and you stop eating it. So this is one cycle of eating, and after like 30 minutes or 2 hours later, your body, your gut digests the nutrition, and it produces the nutrient value, and you have the satiety. So every time you eat something, like the previous speaker said about saccharine eating, your brain makes the memory of your eating. So did you like what you ate? And this memory of your eating changes your behavior for the next time. So you have been eating for more than 10 or 20 years, so your brain have all this memory of what you have ate, and that changes your eating behavior forever. So this is psychology and the neuroscience of eating or any behavior have been studied for decades, and this famous psychology and neuroscience researcher Skinner said that operant conditioning, the consequence of the action changes the probability of the occurring again. So if you like something, you do it more. If you don't like something, and you do less. And her said that there must be a need, like a bodily need, like a energy need, and there is the motivation, drive. So need drives the motivation, and it drives behavior, and when the consequence of behavior, and it restores the need. So it's a need reduction theory. And after several years, Kent Berridge and his colleagues have dissected the wanting and liking of the food or any rewards. So as you indulge in like a drug experience more, your wanting increases while your liking decreases. That's the basis of the addiction. And he further suggests that the wanting for food and the wanting for food is become before you engage in the food consumption, that drives your motivation, and that's appetitive behavior. And as you start to enjoy your food, you experience the liking phase, and then after that you have the learning phase. So that's the concept. And very recently, people say that the reward is two different things. There is the oral proxy reward, that's the pleasure of the food, and there is the another part of the reward that's post-oral, that's the nutrient value of the reward. So I think that the psychology and the neuroscience of eating are evolving as we go on. And when we combine all these things, I think the science of eating can be seen as the state, the energy state of the body, and your brain calculates the need. I need like 500 kilocalories. And what do you want to eat? Do you want to eat bread or pizza or hamburger? And you start to eat your behavior. And as soon as you start to put it in your mouth, you get this very high pleasure. And this pleasure makes you keep you eating for like 10 minutes or 20 minutes. And after several hours, your gut digests the food, and it gives you the nutrient value, and it restores the homeostasis. I think the next speaker will have the full picture about the nutrient sensing and value of the whole thing. And I think this too is the cause of the behavior, and this too is the consequence of behavior. And the brain has these sensory cues that predicts all our sensory experience into the prediction. So I think this is the picture of the neuroscience of eating or the psychology of eating. And the biological part is something that we have been trying to do. So we've been trying to find what is the biological components that regulate the need, and what components, biological components are regulating the motivation for the food. And lastly, we found that there is a leptin receptor and GLP-1 receptor neurons in the dorsal medial hypothalamus that has to do with the need prediction signal sensing. So how do we do this? So we do this in usually using mice. So we think about the psychology first. So there's the motivation. I want to eat something, and I'm hungry. I need something. And is this the food or not? And we come up with neuronal components and how they are connected in the neurobiology, and how do we do how do we know which neurons are regulating which psychology? We use the new neuroscience tools that have strong focus on the temporal dynamics. So if an animal finds food, then it will increase the motivation, and the need for the food will go down, and the prediction of session will go up. So based on this theoretical and temporal theoretical modeling and the biological recording of the neurons, then we can say that some biological neurons are regulating the psychology. So I'd like to have a brief overview of our previous researches. So for this lateral hypothalamus GABAergic leptin receptor neurons, we found that these neurons are activated during the seeking phase and the consummatory phase. And when we activate these neurons, it can drive eating. So I think it shows nicely that the lateral hypothalamus neurons are motivation neurons for the food. And we would like to expand to the non-human primate model. So it makes sense that, so we did the chemodynamical viral activation of this lateral hypothalamus GABAergic neurons, and we could replicate these findings in non-human primates, that we can increase the eating by activating these neurons. And how can we dissociate this need and the motivation? So using computational modeling of what need should be, what motivation should be, and we recorded the neuronal activity of these two neurons, the AGRP neurons and lateral hypothalamus neurons, during this behavior of mouse in a room. When we open the door, the mouse comes out from the room and go and eat a small snack here. And when they finish the snack, they go back. And during this very stereotypic behavior paradigms, we could see that these two neurons are activated in two different temporal domains. So we could say that using the computational theory modeling, that this AGRP resembles the need, and the lateral hypothalamus neuron resembles the motivation. And finally, for this GLP-1 therapeutic the Symposium, we found our recent findings about the GLP-1 receptor neuron in the dorsal medial hypothalamus. So, and to begin with, I think most of you are more familiar with the feedback inhibition. So if you are hungry, you do something, and then later it inhibits your feedback. But there is another important feed-forward preemptive regulation. That's if you see the food, then you react on the food cues. So it's like stopping a car at the stop sign. when do you press the brake? You press the brake even before you get to the stop sign. So by seeing the brake, the stop sign, you start to press your brake so you don't go over your designated target. So you feed forwardly stop your car before the stop designation part. So previous researchers, Yuki Oka and people found that there is feed-forward satiation signal for water and hunger, so gulping and the food cues can temporarily satiate the hunger or thirst even before the ingestion. It has been shown by doing this experiment. So if you give something by oral to the mouth or something to the gut, which one would decrease the thirst or hunger faster? I thought that putting in the gut would make it faster, but on the contrary, putting in the gut decrease the thirst and decrease the hunger slower than the putting in the oral. Even in the foods, even before the oral, when you see the food, this hunger neurons goes down. So this is another proof that there is a feed-forward satiation that happens before the ingestion, and it makes perfect sense that if you are to drink water to quench your thirst, if you control it by your blood osmolality, you will be drinking like 2 liters of water. That's too late because there's too much a delay of the gut absorption. So it's more logical to stop drinking before anything happens in your gut. So this feed-forward signaling has been studied by Michael Anderson and other people. And people say that there are dorsomedial hypothalamus leptin receptor neurons that reacts to the sensory cues that inhibits the HRP, so feed-forwardly to error the prevention, to prevent the overshooting of the eating or drinking too much. Yeah, so that dorsomedial hypothalamus has become one of the feed-forward satiation center, and the Bradford Lowell and other people show that this dorsomedial hypothalamus leptin receptor neurons are upstream of the famous HRP hunger neuron, and before the chow, the eating the food, even before the entering the food into the mouth, this neuron goes up and inhibit the hunger neurons. So dorsomedial hypothalamus has been one of the sensory satiation center for this psychology of eating regulation. And I personally had the chance to test Saxenda for myself. When I injected Saxenda, I felt that even just looking at the food made me full. So even before I start to eat anything, I became so full I cannot start eating it. So I felt this pre-ingestive sensory satiation so strongly. So I wanted to measure this in human clinical trials. So this is the actual clinical trial design. This is a big amount of chickens, fried chickens. And the purpose of the study is to measure how much satiation do you feel. So we give a survey. Do you feel something? Do you feel something? And without the drug injection, usually this pre-ingestive oral phase, when you chew the chicken but don't swallow it, like you enjoy the chicken in your mouth for like several minutes but don't swallow it, no satiation happens. But if you get the Saxenda for several enough, then even before swallowing it, you're just chewing the chicken. You report that, oh, I'm feeling satiated, 72 score. So this is a way to study humans, because human can answer, can say things. So, and in our human brains, we found that dorsomedial hypothalamus in human brains also had the GLP-1 receptor expression, and the mouse too. And we did optogenetics to artificially activate this neuron at our designated time. So these two mice are eating the food at the same time, but when they start to eat, when I activated neurons, immediately they stop eating it. So I can increase this satiation immediately and make the mouse stop eating. So if I do it to you, I can make you stop eating it after 1 minute of your eating. So every time we shine the light, there is complete termination of the eating. How about inhibition? So this mouse, we used these optogenetic techniques to inhibit these neurons. So his friend ate his lunch and left, but he's eating for longer duration. So I think it's not feeling the satiation, so it's eating for longer duration. And when we want to measure the physiological activity of the neurons, so this is the mouse going out of the room and going to the food zone. So for the first trial, this neuronal activity goes up only when the mouse actually eat the food. But when the mouse learns that this corridor, this maze is the road to the restaurant, then as soon as it starts to move toward the food, even before getting to the food, these neurons are activated even by anticipation of the food. And we use this miniature microscope to measure the neuronal activity in a single neuron techniques. So this mouse is going for the famous choco snack approach. So during this approach to the food, these green neurons are activated. But as soon as this mouse starts to eat, this green neuron goes down, and this other population increases. The two different populations we found. So I showed you the Saxenda data of the humans. So we wanted to do the similar thing to the mouse too. So what we did is we injected the cell line and we injected the GLP-1 receptor agonist, and we saw that without the drugs, I showed that these neurons are activated by eating the food. But after injecting the drug, the slope of the increase was much increased. So I think this increase in the neuronal activity slope resembles the increase of the satiation symptom, emotional symptom of the human. So that's why we suggest that this neuronal activity is responsible for this psychological phenomenon. And we did a 3D mapping of the whole the mouse brain to see where they go, these DMH GLP-1 receptor neurons. And there are several targets of the outputs. But among them, we had the privilege to collaborate with Kevin Williams in UT Southwestern. And using electrophysiology and other studies, we could show that these neurons have direct connection to the HRP neuron and inhibit the hunger signal. So we believe that this completes this circuit of the prediction and the need and the motivation in the neuronal circuit. So I think this whole symposium was about to know about the target of the GLP-1. So like about 10 years ago, there were several targets. And last year, some reviewers suggest that there are 3 major targets, and we have been demonstrating this one of the targets. And lastly, I want to conclude that I told you one of my goals was to reverse our human race to a better race. So, but there was the obesity rate are increasing forever, but there was success of decrease in the 2023 by the GLP-1. I think by using these neuroscience techniques, I think we can improve our human race and reverse our human. And we are furthering doing further research using our new techniques to see which neuron population activated which time point. And I think this is my last slide. My goal is to find another kind of therapeutic that can really change the disease cause, not the symptom treatment. So I want to, like the previous speaker showed, that after discontinuing the drug, it has a sustained effect. So if we could find the real cause, and if we can find a real treatment that can reverse this pathology, then I think we can really have the true disease-modifying treatment, not the symptom treatment. That's my goal. Yeah. Thank you for your attention.
Okay, thank you, Professor Hyung Jin Choi. So we will go on next speaker. So the last speaker for this session is the Professor Grexer from KAIST. So Professor Choi is a pioneer exploring how internal state and sensory cue shape his behavior and metabolism. One of like a personal good news for him is that he become a director of Institute of Basic Science, so that he needed to move to GIST. So congrats to him. So today, anyway, he will discuss about the postprandial sensory sensing of nutrient through the microbiome, gut, microbiome, gut-brain axis in flies and mice. Professor?
Thank you. I'm not going to tell you about the GLP-1, but I will tell you about something related to the topic. You can hear me well. So I hope you had a nice lunch. I did. And if you look into your food, it contains carbohydrates, protein, and fat. And as you know, that it's well known that the balanced intake of these nutrients is essential for the well-being of an organism. And so there is a recommended proportion in humans, and it's protein 35, fat 20, and carb 45%. And other animals may have different proportions, like carnivores. Lions and tigers have a greater proportion for fat and protein. But whatever that proportion may be, that they are tightly maintained. So you can imagine that an animal must have a system to detect each nutrient and calibrate how much of each nutrient gets into the body. So first question you may ask is, how would you detect these nutrients? So what is the sensory modality that respond to these nutrients? And as you know, that taste sensing is very important. And so here, taste sensing, I like to call it pre-ingestive sensing. And several labs around the world, including Charles Zucker's lab, have identified taste sensing cells on the tongue that expresses distinct taste receptors that responds to sugar, protein, umami, and bitter, salt, and so on. So what happens is that you have these receptors, and it binds to the ligand in the mouth part, and then it sends the signals to the brain. And then also people have identified, though, what region in the brain is important for eliciting the sensation of a sweet, savory, and saltiness. And so we have a fairly good understanding of the identity of these pre-ingestive senses and their mechanism. But there's a little issue here. Issue is that these pre-ingestive taste sensing can only provide a prediction as to nutrition value of food before ingestion. So you say it's a prediction. It may not be real. Okay? So we can give you an example that some of you guys are drinking coffee and you put a Splenda in the coffee. You like it, but it has no calorie. You like it, but it doesn't do any good to your body. So you've been kind of fooled by this. So another modality that I would like to tell you about is this so-called post-ingestive sensing. So food goes through the esophagus, and it will go through the GI tract, and where ingested food is observed and digested and observed. And there are many nutrients or nutrient sensors in the gut, and it's only begun to be identified in recently. And then once this is activated, then it will inform the brain through the gut-brain axis. So we have two major pathways. One is a vagal pathway, the other is spinal pathway, and also circulation. And then, so still we don't know what region is important in the brain that elicits or respond to the caloric content of a food. Okay? So another difference between a pre-ingestive versus post-ingestive is that the pre-ingestive sensing, they detect the presence of a nutrient. I don't think that anybody knows that if there is any mechanism to detect the absence or deprivation of a nutrient. Whereas a post-ingestive sensing can detect both presence and absence. So let's say if you're deprived of protein, you haven't had protein in diet for weeks, then you're going to have a craving. So the reason why you have this craving is because there is a peptide in the gut that gets secreted in response to deprivation of an essential amino acid, and that inform the brains to go ahead and try to get the protein-rich food. Okay, so today I think I'm going to tell you one story about the sugar. Okay, sugar is sweet and also caloric, and also we know that we respond to sweetness using our taste cells. What about sugar calorie? Can we respond to the sugar calorie? And so years ago, it goes back to 1959. Holman carried out very esoteric behavior experiments to show that there exists taste-independent sensor that detect calorie value of sugar. And but more recently, which was almost 20 years ago, the Ivan D'Arujo, a good friend of mine, when he was a postdoc in Duke, he used these so-called taste-blind mice. So they cannot taste sugar. Okay? And then also we use sweet blind flies. And then we basically give a choice between, so we deprive these animals and then give a choice between L-glucose for D-glucose. D-glucose is real sugar. L-glucose is as sweet as D-glucose, but has no calorie. And you give it this choice, and these deprived animal, they chose D-glucose. So it suggests that there must be another mechanism. There must be a taste-independent mechanism to respond to the caloric value of sugar. So then the question is $64,000. Question is, what is a sensor? Yeah, what is sensor? And it was not easy to screen or identify such a sensor in mice. So we carry out screen in the flies. So throughout the work from my lab for the past 10 years, studied with Monica Deuce, a postdoc in the lab then, we showed that these 6 cells in the fly brain that expresses is a DH44 peptide detect, and it's required for detecting and responding to calorie content of sugar and a direct sugar intake. So when you get rid of these cells here, by the way, this is fly brain, there are only 6 cells, and then these are the processes, and then this process here, they send a long process down to the gut. And when you get rid of these cells, and they no longer respond or detect or respond to the calorie content of sugar. Okay? And then this is the activity of DH44 in responding to D-glucose, and they don't really respond to L-glucose. So, what is DH44? DH44 is analogous or homologous to corticotrophin releasing factor, CRF or CRH. And when I look at the sequence homology, it was fairly high. It's almost 30% identical. So I was kind of thinking that, as you know, that most of this neuropeptide diverges rapidly in evolution. So we thought that this 30% identity is kind of significant. But you know that the CRF has been studied in context of stress for many years, and its role in nutrition sensing has been unclear. So what we did is we decided to look into this. And so CRF neurons are located in the paraventricular nucleus of the hypothalamus, this region here. And then there was the graduate student, Dan, now a postdoc in Caltech right now, and she decided to look into this, and joined by the Shine Kim, was also a postdoc in the lab. And so basically what we did is we decided to do the two-trace assay, similar to what we did with flies. And it was giving real sugar versus fake sugar. The reason why we instead of using L-glucose, we use sucralose. The reason is that sucralose is a lot cheaper than L-glucose. And so we give them choice, and then look at the data. And so choices between sucralose versus sucralose, 0 means that these animals have no choice. And you can see that the fasted animal, they are energy deprived, and they prefer the real sugar. Fat animal, at the beginning, they have a slight preference for sucralose, because the sucralose is sweeter. And then as time passes by, and they develop preference for sucrose because slight preference for sucrose because they were getting hungry. So it looks like we have an assay set up, and then next question we ask is whether CRF neurons in the hypothalamus is important for this behavior. So what we did is we use a optogenetic device, and basically we inactivate this neuron. We can activate them or inactivate the neurons, and then also we use a ligometer to automatically count the outcome. And we found that, so this is the result. Sucrose versus sucralose, and control animals prefer sucrose because they were energy deprived. And then this is an animal in which the CRF neurons are inactivated. You can see that they no longer prefer sucrose. And this is time course. You can see that this is control animal. They know right away that what to get. Whereas this is an animal in which the CRF neurons are inactivated, they can't really tell, and then eventually they prefer the sweeter side. So next question we ask is, so something is going on. Somehow a behavior assay tells me that CRF neurons might be important for responding to the calorie content of sugar. So basically what we did is we we give them food and then decide to carry out the functional imaging of the CRF neurons. So here what we do is we have them exposed to peanut butter, the appetitive cue, and then we are measuring activity of CRF neurons using phybrophotometry. And then you can see that when they are exposed to peanut butter, you see the reduction in the activity of CRF neurons. So this was interesting because I showed you that the CRF is a stress hormone or stress pathway. So people have shown that when you stress the animal, it will activate CRF neurons. And this was a first time carrying out the in vivo imaging on the CRF neurons, and we showed that the appetitive cue can suppress activity of CRF neurons. So this was a significant. Even though this was not like a line of work that we were carrying out, we decided to publish this work. But it begs a question as to whether this reduction is mediated by pre-ingestive taste sensing or post-ingestive gut sensing. Right? So how are you going to address this? So what we did is we decided to carry out the imaging experiments while we are infusing the different nutrients directly into the gut using a catheter. So basically we put the catheter and we wanted to bypass the oral cavity, and we infuse the different nutrients into the gut. And so first experiments, we tried to ensure suppressed activity of CRF neurons, like what you saw with the peanut butter, right? But as you know, the Ensure contains lots of macronutrients, fat, protein, and so on. And then, but at least we know that these, when you give them in the gut directly, it suppresses activity of CRF neurons. So we tested sugar first, and it suppresses activity. And if you look at the time course, within about 2 or 3 minutes, most of activity was abolished, was suppressed. So this tells me that maybe this is how information is conveyed from the gut to the brain is through the neurons, because it's fast. Okay? And then the other experiments we did, so basically we test other nutrients. Well, L-glucose is not really nutrients, but it activated as a CRF neurons. Yeah, so we are kind of wondering why you activate it instead of doesn't do anything. So we tested lipid. Lipid is actual nutrients. Yeah, lipid also activated CRF neurons. So so let me summarize a little bit. D-glucose suppresses activity of CRF neurons when they're fasted. When they're fed, they don't really care. Then L-glucose and lipid activated the CRF neurons. And also, then we decided to check all the nutrients, all the chemicals on a Sigma catalog. And we found that first we decided to test fructose. Fructose is close to glucose. And we test the fructose. It also activated CRF neurons. So we asked, what is going on? Why nearly many nutrients activate CRF neurons, but only one that suppresses activity of CRF neurons were glucose, D-glucose. So maybe I can jump a gun a little bit and then tell you that glucose is the most important fuel, or maybe it's the only fuel for the brain cells. And people have speculated that there must be a glucose sensor or glucose responder in the brain, but people haven't found it yet. So I think we have stumbled upon the actual bona fide glucose sensor in the brain. And so we will get to the question later, but so maybe relevant to this crowd. So how is it this is relevant to obesity and diabetes? So we collaborate with Min-Sung Kim's group, and basically what we did is we made a diabetic mice, and found that now. So you give this is normal mice. We give the glucose, and you see the suppression of activity of CRF neurons. And in control animal, the diabetic model, they don't respond to the glucose. Okay, so I think let me skip this part, and then now question is how go from the gut to the brain? How information in the gut is conveyed to the brain? And I told you there are two pathways: vagal pathway and a spinal pathway. So I sent my students to New York, where I was, and to the Gary Schwartz lab, and so we learned the technique. And it was good to send the students to different parts of the world, and they come back in different person. Their command of English was get better, and then yeah. So basically we cut the vagus neuron, nothing happened. This is control, and then this is a vagotomized animal, nothing happened. When you cut the spinal nerve, this is a control. The spinal nerve, you still see the decrease, but you don't see this immediate, the abrupt reduction in the activity of CRF neurons. So we think perhaps whatever that is detected in the gut goes to the spinal nerve. And I tell you about the LPBN, the lateral parabrachial nucleus, and then I send the information to CRF neurons. Okay? So now then it begs the question is, what if you trace these cells back? Are you going to see the spinal nerve, or are you going to see other like vagus neurons or so on? So basically, we did a rabies experiments. We put not going to go into details about this, but basically what we did is we put the rabies virus, retrograde virus, in the LPBN, and then see where we went. And we see staining, like a discrete staining in the spinal nerve, but not in the vagus neurons. So if you look at this is a spinal nerve, this is head, this is tail, and cell body is like down in the lumbar region, like all the way down in this in the gut. Okay, here cell bodies are here. And then these are all axons. Axon traveling all the way to the brain. So you can imagine we hypothesize that perhaps these dendrites innervating the gut and then collecting information and then sending all the way. So we are kind of surprised by that there's only a single neuron might be able to do so. Okay, so let me, so I think I'm going to just get to that. So why this is important? Why? Well, this is a first elucidation of a function of glucose sensing or glucose responsive neurons in the metazoan. And reason why I just want to give you some historical perspective. And years ago, 70 years ago, more than 70 years ago, John Mayer proposed glucostatic hypothesis. He thinks that onset and offset of a feeding is regulated by glucose level in the blood. And he predicted glucose sensing neurons might be present somewhere in the brain. And then Omura, 10 years ago, published a paper that there exists glucose sensing neuron in a hypothalamus using electrophysiology. Basically what he did is he applied the glucose and then found the cells that respond to glucose. And so I think what we have done is that he didn't really address the glucose sensing in the organismal level. So I think we have brought some marble to the table about in understanding of glucose sensing. Let me show you another function of a glucose sensing neuron. So this is all published, but I just give you a punchline. We talked about insulin and glucagon. Flies do have a insulin analog and a glucagon analog. We call it DELP2. AKH is a glucagon. Punchline here is that we identify a single pair of cell. One cell in one hemisphere. This is glucose sensing. And then a neuron comes out and it bifurcates. This is exon 1, goes to IPC, insulin-producing cells, to promote release of insulin. And this exon 2 goes to this region, and then it inhibits release of a glucagon. So it makes sense. When animal eats, glucose level goes up, then it needs to make more or produce more the insulin, but it needs to block the release of a glucagon. So this is work from the Hyung Jin Choi. He has his own lab in the University now. And this is actual real image. And then what if you get rid of this cell? This is a CN neuron. Then I don't know what diabetes is like in flies, but they get hyperglycemia. So here, when you this is a control, and then when you get rid of it, then they have a high level of glucose in the blood. Okay, and I'm gonna stop. I think I just want to mention that I just moved a few days ago to the Gwangju Institute of Science and Technology, and we're gonna have. So I will be a director in IBS. And also we're gonna found new department called Department of Integrated Neuroscience and Physiology. So we are recruiting in all level.
Okay, thank you, Greg, sir. And also congratulations for your new direction. Okay, so we're gonna stop the first session, and then we will have 20-minute coffee break, and then we will come back on 4:40. Okay, thank you all for participating in session 1. Thank you.